Rajeshri. Good day. I'm a research scholar at MGM University, India. My topic is the potential market of nutraceutical supplements in the onco-wellness sector for its therapeutic benefits. So, uh, actually, as for the WHO has predicted, the number of the new cases of cancer diagnosed every year around the world is said to increase by 69% by 2030 to 21 million. This is only because of the lifestyle changes and the dietary modifications in the world right now going on. So the aim of this presentation is to discuss the current nutraceutical products in the market on natural bioactive extracts, mainly distinguished by a specific polyphenolic profiles. Right now, researchers have already focused on many alternative and complementary therapies, which has already been both by the both have a nutritional values and a medicinal importance, pertinent to the prevention and the treatment of diseases, mainly including natural products with the least or the no side effects at all. So, the requirement, outlook, and the significance of this presentation to give a focused study on this bunch of the nutraceuticals and other dietary products that have been considered as an effective strategy due to their substantial role in the prevention and the treatment of the cancer. So recently, many studies have already been indicated that the cancer is a worldwide health problem that is leading the cause of the death. So what is cancer? Cancer is associated with the changes or the mutation to the DNA within the cell, leading to the damage to the DNA, which cause uncontrolled cell growth, genomic instability, and the tumor formation. So detection of a cancer at an early stage and the proper treatment, it will result in the increased survival probability and also in preventing and the development and the progression of the cancer disease at various stages. Nutraceutical includes the functional foods and the dietary supplements, which are used in the pharmaceutical applications. They have some therapeutic benefits, and the complementary supplements also includes it. And it all used in the prevention and the treatments in cancer and many other diseases. There's a role of the nutraceuticals. It food in terms of the nutrition, we call it the nutrition. It highly contributes to maintaining the healthy lifestyle of a person, which exactly influences epigenic phenomena and modifies the drug response. Many dietary supplements and functional foods, they are known to be a nutraceuticals. They're from the viewpoint of the nutrition. They are the source of both nutrients and the non-nutrients, which contains the carbohydrate, protein, fats, and also prebiotic, probiotics, and some enzymatic regulators. So health and diseases effects are significantly affected by the nutritional supplements, functional foods, and the additional micronutrients because of their ability to slow the development of the cancer cells, stop their proliferation, and trigger the cell death. The nutraceutical and dietary supplements, they use to minimize the side effect that counteracts the benefit of the hospitalized treatments. Specifically, antioxidants and anti-inflammatory activities, as well as the reduction of cell proliferation, have been demonstrated to help the patients receiving the anti-cancer treatment respond better and comply with the treatment regimen. So, do the dietary supplements exactly supports the patients who are undergoing the chemotherapy? So, in cancer, patients undergoing the conventional therapy, the intake of nutraceutical supplements is also very common nowadays because numerous naturally occurring bioactive substances, they're identified in nutraceuticals, have been shown to have a cytotoxic, chemotherapeutic, anti-cancer characteristics. So these all substances, they can kill altered or malignant cells by causing cell growth inhibition, cell differentiation, and changes to the redox state of the cells. But high concentration of these active chemicals might reduce the efficiency of chemotherapy by making the cancer cell less responsive. So in natural nutraceutical products in the cancer prevention, drugs, micronutrients, and the products from the natural sources can all be used to prevent the development of the cancer. Therapy is a course of a treatment meant to cure a medical condition or its precursors, whereas the cancer prevention is recognized as the active efforts to minimize the chance of getting cancer or its recurrence. The main advantage of using these natural nutraceutical ingredients or the products is that they do not have any undesirable side effects. And this is also the main reason for the increasing demand in the market. Now, the herbal-based nutraceuticals and the ingredient-based nutraceuticals. These are two types of nutraceutical products which I have studied. So, first, the herbal-based nutraceutical products in the cancer prevention. It includes the people usually tends to believe in the herbal products because they have minimal side effects and are very pocket-friendly and also chemical-free. Nutraceuticals accompanied by the herbs has an excellent impact on the prevention of different chronic diseases such as the cancer, and sometimes their bioactive ingredients also cure the diseases. Boosting the body's defense mechanism, the immunity from inside, it's called a holistic recovery. It is very essential for the cancer patients, and herbal-based nutraceutical, it is very much useful in this criteria. So specific nutraceuticals are characterized according to their functions, bioavailability inside our body, and the mechanism of action. So these products are important to improve the health of a person, to cope up with the day-to-day challenges such as maintaining the physical health and the stress management, and to increase the activity. At present, the emphasis is on the herbs that may be used both as a food and a medication. Nutraceuticals are now more popular than the medication and health supplements among consumers and healthcare professionals as a result of a progressive action. Now, the active ingredient-based nutraceutical products in the cancer prevention, it includes consuming of biologically active components such as the lycopene or omega-3 fatty acids. They are usually from the fruits and the vegetables we are extracting it. So many laboratory studies have already done on this, which shows the several bioactive food components or the nutraceutical natural items which may help to protect against the cancer. Some examples are the oranges containing the calcium. These are some functional food or beverages examples. Milk with the omega-3 fatty acids, and yogurt with a high probiotic content, tomatoes that have a very high lycopene content. They may be modified in genetically engineered plant-based. Eggs with the higher omega-3 concentrations, etc. These are some ingredients like lycopene, gingerol, vitamin E, that is tocotrienol, tocopherols, oroxylin, thymoquinone, curcumin. These are very much important in the cancers nowadays. So there are some ingredient-based natural nutraceutical products containing of essential fatty acids, omega-6 and omega-3, which are very useful in the contributing to the growth of the prostate cancer cells, which shows a very stimulatory and inhibitory effect in the prostate cancer cell lines. Then the probiotic fiber fructooligosaccharides, which helps support immune responses and immune health as it provides the high quality protein. So these all therapeutic nutritional foods are created specially for those who are affected by or at the risk of the cancer related illnesses. The majority of these items are naturally gluten free and suitable for the lactose intolerance. This is a very important point. The active ingredient-based nutraceutical products in the current market specifically designed for their unique ingredients, which helps in inhibiting the proliferation of the cancer cells, reducing its side effects of chemotherapy, and increasing the immunity of a patient, such as EPA, omega-3, 6 fatty acids, lycopene, gingerol. They are well known for the cancer preventive nutraceuticals, lipoic acid, glutathione, superoxides, dismutase, selenium, copper, magnesium, these all minerals, all functions as a anti-cancer nutraceuticals in the management of a cancer by delaying the apoptosis as seen in isolated cancer cells. Also includes vitamin C, E, carotenoids from the various plants, then essential fatty acids, nanoscale silver particles, and the many mineral-rich blends, protein blends, pure hydro solutions like lycopene, gingerol, etc. So global demand for the nutraceutical. If you see global demand, according to the Global News Wire, the leading player who is dominating the entire nutraceutical global market is in the US right now, which is currently holding at 30% of the revenue share and is expected to grow progressively. in the coming forecast period. In the health supplement growing category due to the high demand in the elderly population. The value of the market size in 2022 will be US for $93.606 billion dollars, and the revenue estimate for 2030 will be around $991.09 billion dollars with a compound annual growth, that is CAGR, of a 9%. So here's the conclusion of my slide. Nutraceutical square measures out within the sort of the isolated nutrients, dietary supplements, functional food, and functional ingredients. They all offer all the essential substances that ought to be gifted during a healthy diet of humans. It provides energy and valuable nutrient to the body that is needed for maintaining the best health. The nutraceutical market is widely utilized in the food and pharmaceutical industry because of the growing awareness of health among the people regarding the use of the nutraceuticals when needed. The industry of the marketing nutraceutical is very much expanding nowadays around the world, and especially in the India has joined that. The Indian people have begun to trust immunity boosting supplements, which has resulted in a significant changes in the consumer behavior. Examples of the flexible shopping habits of healthcare customers, including vitamins, capsules, chewable pills, and gummies. For the prevention and treatment of cancer, the market of nutraceutical products based on active chemicals and those based on herbs, they are increasing. So thank you so much. These are some references.
So thank you. Today I will present a translational study entitled Neoadjuvant Chemotherapy Upregulates HER2 and Neuropilin-1 Conferring Drug Resistance. So I'm Dr. Noura Zahimi from Sultan Qaboos University in Oman. Okay. So we did our study or focus our study on breast cancer because breast cancer is the most, as we all know, it's the most common cancer among all other cancers in female worldwide. And the major obstacles of breast cancer is the locally advanced and metastatic diseases that are the leading causes of cancer mortality. So we all know that breast cancer patients with early stages can survive more than 5 years. However, most of the Asian or the late stage cancer is very common in Asian countries and Middle East specifically. So what we need to do here, just let me, sorry. We just need to, here we go. Okay. So breast cancer has different molecular subtypes based on the expression of different receptors, which are estrogen, progesterone, and HER2, plus of course the Ki67. Okay, so it's subdivided into many subtypes. And the locally advanced patients with different subtypes will receive neoadjuvant chemotherapy, of course, plus other therapies. It depends on their the expression of the different subtypes. So they mainly receive neoadjuvant chemotherapy, and some of the patients might respond completely to the treatment. Some others will not respond or respond partially, and others will not respond and relapse. So the question is, what determines patients respond to chemotherapy? Is it genetics, epigenetics, or molecular signaling? So in our study, we aim to identify molecular changes that involve a neoadjuvant chemotherapy resistance in order to find new therapeutic targets in different breast cancer subtypes. So when we talk about resistance, so we know that breast cancer has a heterogeneous nature with different molecular subtypes and like ductal and lobular cancers, or sorry, ductal or lobular breast cancer. And there are certain population of cancer cells that might acquire resistance acquire resistance. So the resistance can be of two kinds or two types, intrinsic or acquired. So what I mean by intrinsic is the patient do not respond to the initial treatment, or sometimes it can be defined as the patient will not respond to a single treatment. While in the acquired resistance, the patient will not respond the patient will acquire resistance during the treatment or following the treatments, and sometimes the patient might resist multiple drugs, and this is called multi-drug resistance. So to answer our question, what determines patient respond to chemotherapy, and of course we are going to focus on the molecular signaling or molecular profile of patients, of the different subtypes of cancer. we need, first of all, to understand the neoadjuvant chemotherapy setting. So we know that one of the neoadjuvant chemotherapy approach used to treat locally advanced breast cancer patients is a combination of adriamycin and cyclophosphamide, followed by 4 cycles of docetaxel. So a total of 8 cycles, and it's like 3 weeks apart between one period and another, or one cycle and another. So what we have done in our study is to generate resistant variants that mimic the neoadjuvant chemotherapy setting. And we used 2 cell lines in our study, the MDA here, the MDA MP231, which represent the triple-negative breast cancer subtype, and the other one is the MCF7, which represent the luminal A subtype of breast cancer. So I'm going to explain briefly how we generated these resistant variants. So first of all, we will have a plate, like for example, an MDA cells that are confluent. Okay, once the cells are confluent, we will passage them into 3 plates. We'll use 2 plates for RNA and protein extractions, and the third plate will treat it with the first cycle combination of adriamycin and cyclophosphamide for 72 hours. And after, of course, these 2 will consider them, the RNA and protein will consider them as control cells or control samples. After 72 hours, we will wash the cells, we'll wash the plate, we'll let the cells to divide and become confluent again. And once the cells become again confluent here, we'll treat them with the second cycle. We'll passage them first into 3 plates, for RNA and protein extraction, and we'll consider this as the first cycle or samples of the first cycle of adriamycin and cyclophosphamide. We'll continue the second cycle for the third plate here, and we consider this plate as the second cycle of AC. So we'll continue the treatment until we finish 8 cycles. And by the way, here, the whole experiment for the 2 cell lines, it took us around 8 months to let the cells survive, to let the survived cells divide and become confluent again. So at the end, we generated resistant variants from both cell lines that resist 4 cycles combination of adriamycin and cyclophosphamide, plus additional 4 cycles of paclitaxel. We use paclitaxel instead of docetaxel. They are from the same maybe family, but we don't have docetaxel at that time in our lab. So, and we use the IC50 of each treatment based on previous research or previous studies. So now to answer our question, to determine what the signaling pathway or the molecules that cause resistance or induce resistance in the cell lines, first of all, we need to dig into or find molecules that are involved in or confer breast cancer resistance or metastasis. And we found previously in one of the previous studies published in PMC Cancer that neuropilin protein or overexpression of this, when we have done a functional study here, we overexpress this protein, this molecule, neuropilin, which is a transmembrane protein. So we found that the activated pathway after we overexpress this molecule, once the neuropilin is overexpressed, it activates integrin beta-3, focal adhesion kinase, AKT, and the survival NF-κB molecule or marker. Not only in vitro, but also we have found in our previous clinical research that published in Frontiers in Oncology that the neuropilin molecule subtype breast cancer or the tissues of the triple negative subtype compared to the others, and also the tissue of metastatic patients has high levels of this protein compared to the non-metastatic cases. And we also find when we check the survival of the patients, when we have done Kaplan-Meier curve, that the low levels of neuropilin are linked with better patients or better overall patient survival. So we can conclude here this is one of the target biomarker that we need to check in our resistant cell lines. So let's now start with our MDA cells, which represent the triple negative subtype. And we found that when we check the level of neuropilin, this is the control cells or the control samples. These are the cells that resist 4 cycles of adriamycin and cyclophosphamide. The third sample is the cell that resists the full treatment, the 4 cycles of AC plus 4 cycles of paclitaxel. And as you can see, the red color indicates that this protein is highly expressed in these cell lines or these cells. So the neuropilin expression is increased significantly in the resistant cells. Okay, the breast cancer resistant protein has an opposite pattern to neuropilin, but the neuropilin overexpression, it activates PI3 kinase, the phospho-AKT, okay? It's mainly the PI3 kinase, integrin beta-3, which has the same pattern as neuropilin here, and activate the focal adhesion kinase and phospho-NF-κB pathway. So neuropilin induces the survival or stimulates the survival signaling molecules. And here we have, just to check the level of neuropilin or the levels of neuropilin in the control versus resistant cell lines, we have done immunofluorescence. So the green color indicates the expression of neuropilin here. The green in the control here, sample, the expression of neuropilin. It's overexpressed in the 4xAC and the cells that resist the complete treatment. We also checked some of the epithelial to mesenchymal transition molecules, and these molecules are usually the molecules that detach the cells or detach the epithelial cells or the epithelial cells from epithelial tissues and induce their metastasis. So there are two markers of epithelial to mesenchymal transition, which are vimentin and ZEP1 that we checked. And we found that, of course, we checked also E-cadherin, and since the MDA cells are represent the triple negative subtype, you don't express E-cadherin, okay? But when you check vimentin and ZEP1, the two EMT markers, these two are overexpressed as well in the cells that resist the full treatment. So we found, or what we can conclude from here, that resistant MDA cells or triple negative cell line to neoadjuvant chemotherapy activate the survival axis through neuropilin, integrin beta-3, and the NF-κB axis. Plus, it activates the molecules that involve in or induce metastasis. We checked also the other cell line, which is here, the MCF7, which represent the luminal A subtype of breast cancer. And we found an interesting results here. So we have done Western blot here. And the cells that resist the 4 cycles have different pattern from the cells that resist the full treatment here in MCF7 cells. So the cells that resist the half of the treatment upregulates neuropilin, okay? And the TNFR2, PI3 kinase, the proliferation pathway through phospho-AKT, and the phospho-NF-κB, focal adhesion kinase and phospho-NF-κB here. However, the cells that resist the full treatment, they downregulates neuropilin, so they have an opposite pattern now, but they upregulates the other molecule, which is breast cancer-resistant protein, which is again a type of ABC transporter, transmembrane protein, that use sometimes afflicts the drug outside the cell to make the cells more resistant. And also activates integrin here, while the integrin was downregulated in the 4xAC here. So MCF7 has slightly different pattern of, or distinct pattern of resistance compared to MDA cells. Not only that, as I told you before, MCF7 cells represent the luminal A subtype of breast cancer. And surprisingly, we found that the neoadjuvant chemotherapy converted this HER2 negative cell line to a HER2 positive cell line. So after 4 cycles of AC, the HER2 and the active form of HER2 and related HER2 family, which is epidermal growth factor receptor, okay, protein here is overexpressed and it's phosphoform, the active form. So the neoadjuvant chemotherapy activates the HER2 and associated HER2 signaling molecules or associated molecules. Plus, it activates also the estrogen receptor, and we know that estrogen is expressed in the luminal A cells. Okay, so again, estrogen play a role here, which is also upregulated in the resistant cells. So now, to determine which drug cause the conversion of the MCF7 from HER2 negative to HER2 positive, we have done a single drug treatments like adriamycin and paclitaxel for a short period ranging from 2 hours to 48 hours here. And surprising, and of course, we checked for neuropilin, HER2, and some epigenetic molecules, histone deacetylase 4 and histone deacetylase 3 here. So we found that we checked for neuropilin first, and we found neuropilin is directly activated after 2 hours of the treatment with either drug. The same pattern was observed in HER2 here. So HER2 was directly overexpressed in the after 2 hours with either of the drugs. The active form of HER2, the force of HER2, was activated at very late period. When we treated the cells with cyclophosphamide and adriamycin, but when we treat the cells with paclitaxel, it directly activates HER2. The histone deacetylases or the epigenetic markers had similar patterns or have similar patterns to the neuropilin and HER2 here. And just to confirm the expression of HER2 and neuropilin, because these two are the main molecules in our study, we have done dual immunofluorescence staining here, and we found that, so this is a control cell, and this is the cells that resist 4 cycles of AC, and here we have the cells that resist a complete 8 cycles. So this is a DAPI stain, the nuclear stain, HER2 stain, HER2 antibody, the neuropilin here, and here's the merged images. HER2 directly induced after 4 cycles and after the complete treatment. Neuropilin was overexpressed after 4 cycles, but downregulated later, slightly reduced here. And these are the merged images of these of the dual immunofluorescence staining. So now, when since we observed, like in the single drug treatment, that neuropilin and HER2 having the same pattern, so now we'd like to check whether overexpression of HER2, if we overexpress HER2 in the luminal A cells and the triple negative cells, is the HER2 going to induce the expression of neuropilin or not? So are these two proteins or molecules working together to induce metastasis or induce cancer cell survival or not? So we have done here a gateway cloning to overexpress HER2. And here are the results of the overexpression functional study that we have done. So we have here the MCF7 cells. And here we have the MCF7 cells that overexpress HER2. We call them MCF7 HER2. And here we have the other cell line, which is the triple negative cell line control. And this is triple negative cell line that overexpress HER2 through this functional study or gateway cloning. As we can see here, the HER2 unassociated molecules are overexpressed in the HER2 overexpressed. So this is what we expect to have, right? Because we overexpress this protein in the cells. We genetically engineered these cells, so we overexpress HER2 in them. So in MCF7, we found that overexpression of HER2 also induced the expression of estrogen receptor. So estrogen was also high here. Neuropilin was also overexpressed in both cell lines here. Of course, the ER is not present here because this is triple negative. That's do not present or do not express estrogen here. While neuropilin is overexpressed in both cell lines, which means that overexpression of HER2 will induce the expression of neuropilin as well. Plus, of course, if we check here the survival axis, it's also overexpressed once we overexpress HER2. We also check we have done spheroid assay to check the size of the spheroids or the 3D cells when the cells form a 3D. Okay, and here we have the MCF7 control cells, and this is the HER2 overexpressed MCF7. You can see the difference in size, the size of the spheroid between the control and the HER2 overexpressed cells. MDA cells, they lack eco-adherent and they do not form a good spheroid, but sometimes they form mammospheres. And as you can see here, the number of mammospheres there's a difference between the HER2 overexpressed MDA and the HER2 negative MDA cells, the control cells. So again, we have done dual immunofluorescence to check the co-expression of the two molecules, the HER2 and neuropilin. And here we have the HER2 positive MCF7. You can see the HER2 is overexpressed here. The same for MDA cells, the HER2 was overexpressed here as well. Since MDA cells or triple negative cell lines overexpress neuropilin, okay, we can see now the difference in neuropilin level between the control and the HER2 overexpressed cell line. And here's the merged image. So both, when you overexpress HER2, the neuropilin will be also overexpressed. So this lead us to a question. Are these two proteins interacting together? Are they binding together to enhance cancer progression? Okay, so we have done co-immunoprecipitation experiment. So it's basically, we will have a beads. We attach the bead with the antibody. For example, here, in our study, we have attached the beads with the HER2 antibody. So now the target protein is the HER2. So once we add the protein, the HER2 will bind directly with the antibody. Any protein associated or binds HER2 will directly bind when we bind the proteins attached to the beads. So we co-immunoprecipitation, we pulled HER2 and we checked for neuropilin in Western blot. And surprisingly, it's there, which means that neuropilin and HER2 are binding together. I will not express here, this just to show the normal levels of HER2 and neuropilin in different breast cancer cell lines. So just to confirm this binding, we just want to make sure that these two proteins are binding. We have done also the co-immunoprecipitation in the overexpressed or HER2 overexpressed cell lines. And as I told you, since MDA cells overexpress neuropilin, it's very obvious the binding between the two. So again, we immunoprecipitated HER2 and we checked for neuropilin in the first day, and it's there. It's very high in MDA HER2 cells, or MDA overexpressed HER2, or MDA cells that overexpress HER2. And then the second day we checked for HER2 antibodies, and it's there. So it means that these two proteins are binding together. So these two proteins work together to induce cancer progression or induce resistance. So we have here, just to conclude here, that different breast cancer subtypes show different, or the two cell lines, or the two cell lines that represent the triple negative and luminal A have distinct pathways of resistance. So A cells overexpress neuropilin, EGFR, integrin beta 3, to activate the survival axis here. While cells resistant to MCF7 cells that represent the luminal A, that resist 4 cycles of AC, it activates so many molecules, should be also assessed in patients undergoing chemotherapy resistance. So at the end, since we have anti-HER2 therapy or targeted therapy, it's also important to use anti-neuropilin therapy as a targeted therapy that might increase the treatment success and improve patients, or presented today are published already in British Journal of Pharmacology. If you want to go through the results, and there are more experiments are here, just presented half of them, or maybe less than half of the results. So finally, we'd like to acknowledge all the people involved in the study, Dr. Serene Adham and Dr. Mahmoud Yaish from the Department of Biology, those people from our colleague from Sultan Qaboos University Hospital, and of course, there's a great appreciation for the Research Council in Oman for funding this project. So this is the end of my presentation.
Afternoon or evening, wherever you are in the world. My name is Zaliha Al-Marzouqi, a senior science teacher from Oman College of Health Science. And today I'm going to present about my study, which is about the knowledge, attitude, and practice of Omani women on breast self-examination in Oman. And in this study, I use the approach of constructivist grounded theory. The content outline of this presentation will be as the following. First of all, I will touch briefly about breast cancer and breast self-examination, the rationale and justification of the study, along with the aim and objective will be elaborated, study method will be discussed, and finally, the result and conclusion section will be explained toward the end of this presentation. Breast cancer. Breast cancer is considered one of the most health-threatening and is considered the top cause of morbidity and mortality among women. Breast cancer is the second leading cause of death in the world, and in the Middle East, and especially in Oman, it is observed that breast cancer rate is increasing. According to Ministry of Health in Oman, in 2022, breast cancer is the second leading cause of death, and it accounts for 32% of the total registered cases of cancer. Breast cancer is present at young age with advanced stage among Omani women. Breast cancer is the type of cancer that usually faced by appropriate health education. Knowing the risk factors, the signs and symptoms, the ways to detect breast abnormalities, obtaining early nursing and medical care all improve the survival rates of women. There are many research studies show that increased mortality rate of breast cancer could be due to lack of knowledge about breast cancer and its early detection and management. Also, women attitudes and practice of breast self-examination play an important role in discovering breast abnormalities before breast cancer spread. There are many studies show that factors such as knowledge among women and healthcare professionals, professional recommendation, socio-demographic factor, beliefs, religious, family, and social support availability of the service all impact women's behavior toward obtaining medical advices or treatment. In Oman, the Ministry of Health provides many ways to detect breast cancer. For example, they provide treatment for women with breast abnormalities in many secondary and primary healthcare institutions. Also, the Ministry of Health supplies all healthcare institutions, especially the primary healthcare care institution, with mobile mammography buses during Breast Cancer Awareness Month to discover breast abnormalities among women. One of the easiest ways to identify breast abnormalities is conducting a breast self-examination. This practice would help the woman to detect any abnormalities within the breast. However, the usage of breast self-examination among the Omani young women or Omani women in general stays low. Research problem. Up to knowledge of the researcher, there is no research study conducted in Oman examining knowledge, attitude, and practice of breast self-examination among young women and their mothers. There are few studies explore the perception of women with breast cancer. Thus, this study is very important in order to understand what is going on in Oman regarding breast self-examination from the point of view of young women and their mothers. This study would help to understand Omani women thinking about breast self-examination and if they are really practicing breast self-examination. The rationale and justification of this study, knowing what is going on in Oman from the point of view of young women and their mothers, knowing the attitude and whether they are practicing the breast self-examination would help in developing a program about breast self-examination in Oman in order to educate women with appropriate age to do breast self-examination and also to develop women's skill in conducting breast self-examination. There are 3 questions related to this study. The first one is, what do young women and their mothers know about breast self-examination? What is the attitude of young women and their mothers toward breast self-examination? And the third question, what is the practice of young women and their mothers about breast self-examination? The aim of the study was to explore the perception of young women and their mothers regarding to breast self-examinations, and the objectives were to explore the knowledge, attitude, and practice of young women and their mothers about breast self-examination. For this study, we selected to use qualitative approach. And as you know that qualitative study is a process of examining and interpreting data in order to elect meaning, and it will help us as a researcher to gain understanding and develop empirical knowledge. It will help us also to gain an in-depth insight from people who experience the phenomena, which is the breast self-examination here, and allow us to get an inner experience of the participant to determine who meaning are formed. And for this study, we use the approach of constructivist grounded theory. because it is a set of rigorous research procedure leading to the emergence of the conceptual categories. It helped to generate a theory which is grounded in the data, and also it helped to examine all possible theoretical explanation of the initial finding. And for this study, we used, as you said before, qualitative design using constructivist grounded theory to explore knowledge, attitude, and practice of breast self-examination among young women and their mothers. Here we spoke with the participant to interpret their words regarding breast self-examination. And the work of this study was based on the interpretive method because it focused on how the participants' social experience were understood. The ontological aspect of us as a researcher, which was based on social meaning, which considered that word contain individual who have their own meaning. So what we believe that there are many realities or many truths rather than one reality or truth. Epistemologically, we have like a constructivist stance because we do believe that reality is socially constructed through the interaction between us as a researcher and the participant. This methodology is very important and help us as a researcher to build a theoretical understanding of breast self-examination by understanding how participants perceive their world through social interaction. The setting of the study, this study was conducted between February and to October 2023 in North Batna Governorate of Oman at 3 secondary girls' schools in Sohar and Saham wilayats to reach young Omani women and their mothers to explore their knowledge, attitude, and practice of breast self-examination. And North Batna Governorate was selected because it is the second governorate in Oman by population density. The sampling for this study, purposive sampling, which based on inclusion criteria was used to recruit participants for this study. And of course, we consider variation while recruiting the participant. For example, various age group, various residence place, various schools, and various levels of education. At the beginning of data collection, purposive sampling was used, and it was relies on the following criteria: young women studying at secondary school, mothers and their daughters living in North Batna Governorate. If the young mothers were unable to participate in this study, then the young women will be excluded from this study. In this study, both purposive sampling and theoretical sampling techniques were used, and the theoretical techniques here help us to select whom among the participants can give more information, and this will help to develop the theory. Regarding to sample size, in grounded theory, the exact number of the sample cannot be identified before conducting the initial data collection and analysis. So it is advisable for the researcher who are using grounded theory that the sample size is developed inductively until we reach to the point of data saturation. In the current study, we were adding participants until theoretical saturation was obtained and no new information were emerged from the data. In this study, 30 participants, 15 mothers and 15 daughters were interviewed and considered to be enough to reach to the saturation point. Regarding to the unit of the study, for this study, we recruited 30 participants and we conducted interviews with them, 15 mothers and 15 daughters. Regarding to the mothers, they were married and the age ranged between 40 to 55 years. 80% of the mothers obtained secondary school certificate and 20 had higher degree certificate. 73.3 mothers were housewives and 26.7 were working mothers. Regarding to the young women, They were all the daughters of the same above mothers who we did an interview with them. And we did interview with them on the same time that we did with their mothers. And the age group ranged between 16 to 18 years. And they all were studying in secondary schools at North Batna Governorate in Oman. Regarding to the recruitment, the participants were recruited via poster advertisement, which were hanged at different schools at North Batna Governorate. The participant information sheet describing the purpose of the study was given to the participant who were interested in participating by recruiting person. The contact detail of the potential participant and oral consent for the researcher to contact the participant were obtained by the recruiting person. Then the recruiting person give the information, which is the contact number, to the researcher, and the researcher contact the participant via phone and answer their question. Then they agreed on the time and place to conduct the interview, and informed consent were obtained before the process of data collection. Regarding to data collection methods and tool, for this study, we talked to young women and their mothers to interpret their point of view regarding to their knowledge, attitude, practice of breast self-examination. Also, we discussed with the participant about the issues related to practicing breast self-examination to understand their knowledge, attitude, and practice about breast self-examination in more detail. Before starting the interview, we have developed an initial interview topic guide, which was based on a few questions. And these questions were open-ended questions related to breast self-examination. For example, what do you know about breast self-examination? The topic guide was used to ensure that the relevant questions were covered to meet the aim of the study. Discussion with the participant allow us to explore the participant views on breast self-examination. And after a few interviews with the young women and their mother, it was clear that question needed to be asked to the topic guide in order to understand further issues that were emerging from the conversation with the young women and their mothers. Thus, we amended the interview topic guide accordingly. And this study also used semi-structured interviews, and we consider this type of interview as a very important method to collect the data regarding to participant knowledge, attitude, and practice of breast self-examination. Initially, semi-structured interview were conducted with 6 participants, 6 mothers and 6 participants, 3 mothers and 3 daughters. The initial analysis of the data show the need to recruit additional women to the study. And as you know, flexibility within interview is very important to explore a new idea about breast self-examination. Using this type of interviews allow us to follow concern related to research question, explore and clarify comment made by the participant, and therefore this kind of interviews allow us to ask question in the same way, but with great depth. Data processing. Interview were anonymized, and we coded each participant, and also we revised the transcript for accuracy. We do audio record all the interview and transcript all the data. Also, we made sure that to record every single word of the participant to make sure that the result developed from the data, not from our own personal view. We try to maintain credibility of this study by trying to establish a true picture of the phenomena under the study, And also we allowed certain transferability of the study by providing adequate detail of the context of the field for the reader to decide whether the main environment is similar to another situation with which they are familiar, or whether the result of the study can be justifiably be applied to another setting. Also, we tried to achieve comparability, in which we took a step to determine that the result developed from the data, not from our own predisposition. Thus, we considered ourselves as qualified to understand the participant knowledge, attitude, and practice about breast self-examination. Regarding to data analysis, we analyze the data using constant comparative analysis. And as you know, in grounded theory, data collection and data analysis occur at the same time. And thus we use theoretical sampling to feed constant comparative analysis of the collected data. At first, we used open coding to create categories from the collected information, and within each category, we tried to establish several subcategories. Coding in this phase helped us to shape the analytical structure of the collected information. And during this type of coding, the goal was to stay open to all possible theoretical direction indicated by the data. This step in coding encouraged us to decide about defining the core categories as well. And then we moved to line-by-line coding, in which here we read the data systematically, and then data were coded. Each line were coded by specific data or code. Also, we tried to compare data to find similarities and differences, and also compare the data with each other to generate conceptual categories. And these all in the first steps of data analysis. The second step in data analysis is focused coding, and here the researcher analyzed the data in another way, or in a new way, by pinpointing the most outstanding categories from the earlier codes, which we did in the first coding. Focused coding provides us with a concentrated way of looking to the data, and it helps us to gain a deeper understanding of the realistic world of the participant. Of course, we considered the ethical consideration for this study, and the approval of the study was obtained from Ministry of Health Ethical Committee in the Sultanate of Oman. Before conducting the study, the design and the purpose of the study were discussed with the Nursing and Midwifery Department in the Directorate General of Health Services in North Batina Governorate. The reason was to get permission to use study allocations, and also to get all facilities to perform the study. Also, the purpose of the study was discussed with the directors of the selected schools in order to hang the advertisement poster. Regarding to the result, as we said before, the data from the interview underwent constant comparative analysis, and two main categories were emerged. The first one is constructing knowledge and attitude, and the second one is practicing breast self-examination. Regarding to the first category, the data revealed that age and the marital state were significant for knowledge about breast self-examination. Surprisingly, less age and single women were more knowledgeable about breast self-examination than older and married women. The young women, I mean here the daughters, were more knowledgeable about breast self-examination, the importance of it, than their mother. Also, the young women were more knowledgeable about the appropriate age to conduct breast self-examination, the timing, and the practical aspect of breast self-examination. The reason for that was the young women constructed their information about breast self-examination from a well-educational system, whereas the mothers and majority of mothers constructed their information about breast self-examination from the older generation, the grandmother, I mean here. Therefore, mothers perceive that breast self-examination should be performed when the mother or woman reach 40 years of age or above. The data also revealed that mothers with high level of education were more knowledgeable about breast self-examination, appropriate age, the timing, and also the practical aspect of breast self-examination than the mother who got only secondary school certificate. The data also revealed that there is no significant relationship between employment and participant knowledge of breast self-examination. Regarding to the second theme or category, which is practicing breast self-examination, this category explored participant practice of breast self-examination and if they were regularly conducted breast self-examination. According to mothers, they learned from their mother, I mean the grandmother here, that breast self-examination is not necessary if women don't reach 40 years of age. Also, other women were not practicing breast self-examination because they did not have a family history of breast cancer. The busy life of mothers lead to forgetting to conduct breast self-examination at an appropriate time, and the majority of mothers did not trust their ability to do or to practice breast self-examination. As a result, they did not conduct or perform breast self-examination. Regarding to young women, they were confused regarding practicing breast self-examination, though they were knowledgeable, but they did not practice breast self-examination because they received information from two trusted sources. The first source was their mother, and the second one, educational system. The first source, their mother, were insisting that they don't have to do or practice breast self-examination. And the second source, which is the educational system, give opposite information to do breast self-examination at the age of 20. Thus, they were confused whether to practice breast self-examination or not to practice breast self-examination. In conclusion, this qualitative study explored the perspectives of young women and their mothers regarding breast self-examination. In sum, the mothers were lacking knowledge about breast self-examination, and also they were lacking knowledge about how to conduct breast self-examination. Both group, the young women and their mothers, have positive attitude about breast self-examination, but the mothers were not practicing breast self-examination, and majority of mothers receive information from the previous generation, and they reported that they did not practice breast self-examination due to following reason: age less than 40, no family history of breast cancer, busy life which lead to forgetting, and inability to do breast self-examination because according to the mothers, they did not receive any information or any practical aspect regarding to how to conduct breast self-examination from the healthcare professional. The young women, though they have good information about breast self-examination, but they did not conduct a breast self-examination because they were confused, as they received information about breast self-examination from two trusted sources, and they were not practicing breast self-examination. This is the end of my presentation.
Good morning, good afternoon, and good evening to all the participants here. Myself, Dr. M. Sasikala, M.Pharm PhD, Professor and Head Department of Pharmaceutical Analysis, Katwa College of Pharmacy, Coimbatore, Tamil Nadu, India. I'm very happy to present my research work in this wonderful international webinar on women's health and breast cancer today. My title is Research on Herbal Drug Development for Breast Cancer Treatment in Women. I'm very much interested to do research in herbal drug since many of the allopathic drugs cost more. Based on the cost and side effects of allopathic drugs, my research is focused on herbal drug development for breast cancer treatment. Due to the breast cancer, many cases found as per the status in India and also in the world. My focus is on breast cancer treatment to develop a drug for the same disease. The contents of my presentation: introduction, abstract, methods, results, outcome of the research, relevant publications, online research communication profiles, acknowledgement. First, let's move on to introduction on the research. Many have discussed about the global status of cancer. It's absolutely more than 20 million people are suffering from cancer. Among that, around many people are suffering from breast cancer. In that, triple negative breast cancer, TNBC represents triple negative breast cancer, which is more prevalent comparing to all other cancers, and it is more spreadable. It is due to ER, PR, and HER2 receptors, estrogen, progesterone, and human epidermal growth factor 2. When the absence of these three receptors causes triple negative breast cancer, and also proteins called E-cadherin, when the lack of these receptors and proteins causes triple negative breast cancer, and which may cause metastasis because of that cytokines level, metalloproteases level, kinases level, VEGF gene level, STAT gene level, everything will be increased, and it automatically causes reduced programmed cell death, that is apoptosis. So everything leads to triple negative breast cancer. This is the major mechanism involved in developing breast cancer. Now let's look on to the abstract of my research work. I have chosen the plant that is Ipomia aquatica. Ipomia aquatica local it is the local plant found in India, and the name of this plant, vernacular name is water spinach. And from this plant, which will be having the differentiation between other species, the flower has white along with purple color. So from this we can differentiate the species of Ipomia aquatica with other species. Others also have many effects, and many articles were reported, many research books were reported on other species comparing to Ipomia aquatica. And I have chosen this plant, and from this I have taken extraction, that is of hydroalcoholic extract. From that a fraction was isolated through column chromatographic method, and this fraction I took because of high yield everything. I have taken this fraction for antioxidant studies and also in vivo anti-cancer activity studies, since it has antioxidant effect on ROS species and it inhibits PGP efflux and MDR multi-drug resistant proteins, multi-drug resistant associated proteins 1 and 2, everything it inhibits, and also translation growth factor alpha and beta it inhibits, so that cell proliferation, angiogenesis, all the process will be inhibited, because the fraction might contain polyphenolic groups, mainly glycosides. I found it is a meramoside resin glycoside. It is a variety of resin glycoside. It was already published on this compound, and it is structurally characterized, and it has been published. Now I am discussing only in vivo studies on the isolated fraction from Ipomia aquatica. Let's see the methods what I have carried out for in vivo studies. Firstly, I have collected plant collection, extraction, and isolation. Plant was collected in the local area of Tamil Nadu, and it was identified by Botanical Survey of India, Coimbatore. And after the collection of plant, I went for extraction and isolation through column chromatographic method. And the selected extract was hydroalcoholic extract. And from the extract and isolated compound, both were taken for in vivo anti-cancer evaluation, where I have used DMBA-induced method. Before carrying this in vivo method, I have already carried out all preliminary characteristic studies like TLC, HPTLC, all identification tests, pH test, and also leaf studies, pharmacognostical studies on the leaves, which I have selected. And then I have gone for MTT assay method also using MCF cell line. Finally, I went for in vivo anti-cancer evaluation using DMBA induced method. DMBA is nothing but dimethyl benzanthracene, a chemical reagent causing cancer, breast cancer in animal, which I have selected. Animal used was female albino mice. 6 groups I was selected. 6 animals per group was selected, and it was obtained from for using animals. I have got approval from IAC committee in the year 1920. And after by using animals, I have carried out breast cancer study by the method DMBA induced method. What are the parameters I have studied was hematological parameters, biochemical parameters, and histopathological studies. Finally, statistical studies. Let's see the results for all the studies. So after inducing breast cancer in the selected animal, that is all female albino mice, I have selected 6 groups where each group contains 6 animals, 6 animals per group as per OECE guidelines. And I have selected 5 groups. One of the group I have done for another compound also. Isolated compound, 2 compounds were selected, but now I am here focusing only one compound, which was characterized completely. So here 5 groups. G1 treatment animals are divided into 5 groups with 6 animals, and the G1 kept as negative control, and G2 was positive control, G3 was standard, G4 was extract, that is hydroalcoholic extract of plant, and G5 was isolated compound. So comparing with the negative control contains only solvent, G2 contains tumor control, that is positive control induced with DMBA, G3 was treated with standard used was vinblastine, and G4 was extract, that is the plant extract, hydroalcoholic extract of Ipomia aquatica, and G5 was isolated compound, that is resin glycoside. So first, let's see what are the hematological parameters studied. WBC, RBC, hemoglobin, packed cell volumes, and percentage packed cell volume, and platelets. WBC were analyzed per cells per milliliter into 10 to the power of 3, and RBC cells count were measured in millimeter cube, and what a hemoglobin content was measured in gram per deciliter. This is in percentage, and this was measured lakhs per cubic millimeter. So as per the results, where negative control shows for WBC increased level, since few cancer cell lines will show increased WBC content, which causes leukemia. So that our drug molecules are bringing the WBC level to normal from 14 to 11. So the values are significant when it was analyzed statistically with ANOVA test, one-way ANOVA test. And RBC count, let's look onto the RBC count. It was decreased with a negative control. The group 2 was negative control, positive control, that is tumor control, tumor induced, where RBC cells are reduced. So our drugs are comparing to standard extract, G3 standard, G4 extract, G5 isolated compound. These 3 are increasing the levels of RBC significantly. Likewise, hemoglobin content also were decreased and in tumor control. So with respect to tumor control, our drugs are increasing hemoglobin level. Packed cell volume, percentage cell volume was increased and it is bringing to normal by the drugs. Platelet counts also diminishing and it is increased by the drugs with the anti-cancer properties. Now let's see what other studies were carried out. That is biochemical studies for using enzymatic antioxidants. The enzymatic antioxidants used were, which is related to prevention of cancer cell growth. These enzymes will play major role in preventing tumor growth. So if you check the levels of enzymatic antioxidant, you will come to know how much our drug is acting on it. So SOD represents superoxide dismutase and catalase, glutathione peroxidase, lipid peroxidase. These four enzymatic antioxidants were studied. So as per the study, the result shows all three enzymes were decreased with tumor control. When tumor is induced, superoxide dismutase, catalase, and glutathione peroxidase, these three levels are decreased, where lipid peroxidase level is increased because their free radicals level is increased. So this causes increased tumor growth, where other enzymes are decreasing in when tumor cells are increased. So our drug aims to increase the level of SOD, catalase, and glutathione peroxidase. What these enzymes will do, it will decrease the free radical formation, that is ROS species, which we have seen in the introduction slide itself. So ROS reactive oxygen species will be decreased by this enzymes, which causes a decrease in tumor growth. But when we increase tumor growth, it will be decreasing. So it causes tumor proliferation. So to decrease that, our drug should act on this particular enzymes and it will increase the level of these three enzymes, whereas it should decrease lipid peroxidase. So significantly, it shows the results. So when comparing with the tumor control, you can find the sample selected, isolated, and the extract obtained from the plant. And comparing to the standard vinblastine, it shows significant result in improving the anti-cancer effect. And this is the histopathological study, shows group 1, group 2, group 3, group 4, group 5 animal tissues. And the first set shows normal control, tumor control, and the standard activity of standard on the breast tissue taken from female albino mice. And the second set shows here normal control, tumor control, and extract activity. You can find the decreased cell growth here. And third set shows normal control. Normal control is negative control, and tumor control and isolated compound. So you can find standard, extract, and isolated compounds are showing significantly decrease in cell growth level in the breast cancer tissues of female albino mice. And let's see the tumor growth. And tumor growth was measured by means of three factors. One is body weight was measured, and tumor volume and percentage reduction of tumor was determined. The body weight, absolutely it is decreasing. In negative control, tumor control G2 group, it was decreased because of tumor body weight is reduced. So our drugs are trying to increase the body weight of animals. So you can find the drastic change and significant change comparing with the tumor control when in the treatment groups, where tumor volume also is decreasing significantly with the treatment groups comparing to tumor group. Whereas reduction of tumor also significantly shows increased for standard, whereas extract and isolated compound. And the statistical significance of this results were analyzed by one-way ANOVA using Newman-Coles multiple range tests. Okay, so according to this test, all the values were significant comparing to the controls used. So this is about the research. From this research, the selected isolated resin glycoside from Ipomia aquatica has been evaluated and which shown better anti-cancer activity. Further, the research is going on developing nanoparticles. Among many nanoparticles we have tried, silver nanoparticles are showing better results. And it will also work for meeting out multi-drug resistance, which is caused due to allopathic cancer treatments. Many tamoxifen and other drugs, methotrexate, other drugs are inducing multi-drug resistance. So this will also overcome by this selected isolated glycoside myrmoside. So furtherly it is proceeded, and we will carry out many research on it, and we will bring surely from our institution, with the support of our management, we will bring a novel herbal drug in future for treating breast cancer in women. So definitely it will meet global requirement for new drugs in treating breast cancer in women, which is a common immunological disease found among them. So related to this, relevant publications I had from 2012 onwards. So I have studied this plant in various angles, and I have published also the isolated compound with multi-drug resistance activity, and then I have published another molecule also, and defensive consequences by using DMBA method and HPTLC studies. All other evaluation parameters were published, and I have studied with another species of Ipomia, that is obscura. Then aquatica with a different MTT assay also was published during 2020, in the year 2020. And then the molecule, I said two molecules I was isolated. The first one also was published in the Research Journal of Pharmacy and Technology, and many characterization studies in order to determine the chemical structure of myrmoside. I have carried out studies, structural elucidation studies, where I have published FTIR, and even the isolated compound study. When you go through my articles, you can find how to predict the chemical structure using FTIR, NMR, and mass spectroscopic methods. And many HPTLC methods using other plants also are in progress. Peristylus puderoides, another plant, and can find many works carried out through Ipomia aquatica. And Eugenia jambulana for anti-cancer property, anti-diabetic properties also I have studied. Curcuma cassia. So these are many relevant publications on herbal drug development and relevant research about which I was carrying out, and I am carrying out still, and it is continuing to develop a herbal drug. My aim is to develop a herbal drug for definitely for breast cancer. Being a woman, I at least I have to develop a single drug for treating breast cancer without any side effect with a low cost. That is my aim. So it is under progress. And I have it is for participants whoever presented very nicely. Really I'm very much interested to listen to all your presentations. It is a different angle you are doing research, and you can hope you will be knowing if new researchers are there. You can find many communication profiles, research communication profiles, so that you can extend and you can share your research books with many colleagues. So here is ORCID ID. Hope everyone knows. Whoever is doing publications, you can create ORCID ID. If anybody is not created, you just create ORCID ID, where you can find Publiance ID and Scopus ID. Also, you can interlink like Aadhaar and PAN, how you are linking. Like this, you can link all other research IDs, and it will be automatically updating your research updates in the profile when you create. And this is Scopus profile of mine, and this is ResearchGate profile, and this is Google Scholar profile, where you can find your citations, H index and ITIN index, and which is created using institution mail ID. And Web of Science, that is Web of Science researcher ID through Publiance, we can register it, and here also we can update it, and it is also linked with ORCID ID. And through this Web of Science profile linkage, you will get many reviews, editor options, and you will get options to publish in Web of Science journals also your research works. And it is academia profile. Recently I have created this, so I have not uploaded it in this profile. And these are a summary of online research profiles. So I'm concluding with the proverb: research is creating a new knowledge. It is said by Neil Armstrong. And I acknowledge our principal, Dr. S. Mohan Sir of Katpam College of Pharmacy, Coimbatore, and management of Katpam Group of Institutions, Coimbatore, for giving me this opportunity and support to carry out a vast research on cancer studies, and organizers of this conference for giving me this wonderful opportunity to present and share my works among many wonderful researchers and supporting. I thank my family, colleagues, and friends for supporting me in all the ways. And from all, the grace of God is with me for presenting every time. Motivating me, supporting me, mental support was given by God Muruga. I thank you all for listening patiently for my presentation. Laughter is the best medicine in the world, and every disease a herb to cure it. So we will together develop a herbal medicine for breast cancer, treating breast cancer. Thank you all.
Everyone, I would like to thank the organizing committee of the second international webinar for accepting my intervention. My presentation is titled Parvovirus B19 Infection in Pregnancy, Clinical Manifestation, Diagnostic and Adverse Fetal Outcome Systematic Review. The next slide. The next slide. Some maternal infections contracted before or during pregnancy can be transmitted to the fetus during gestation, during labor and childbirth. The agents responsible for this infection can be viruses, bacteria, protozoa, among viruses most frequently responsible for congenital infections are cytomegalovirus, CMV, herpes simplex, zoster virus, and parvovirus B19. Parvovirus B19 is a non-present pathogen and contagious small viruses discovered in blood donation bug bearing reference B19. B19 virus is a human pathogenic virus belonging to the erythroparvovirus genus in the Parvoviridae family. Parvovirus B19 characterized by a marked tropism of erythroid progenital cells. The pathogenic role of the virus was identified during erythroblastopenic attacks in patients with sickle cell disease. B19 causing a broad spectrum of clinical manifestations: childhood rash erythema, emphysema, arthralgia, and in pregnant women it can cause fetal death, spontaneous abortion, or hydrops fetalis. Acute parvovirus B19 infection is a risk for pregnant women. Close and frequent contact with young children is a major risk occurring B19 infection during pregnancy. The other risk factors are working in the school, care centers, and other full stress jobs. Primary infection manifests as rash, causes a sloppy cheek, and arthritis. B19 can also be transmitted vertically if a person is infected during pregnancy, with 26% to 44% of pregnant individuals susceptible to infection. The maternal risk of infection is high during the first trimester. However, the fetus is at greatest risk of infection during the second trimester. Studies have shown that 1 to 3 weeks following maternal infection, the fetus is infected in 17 to 35 of cases. B19 infection during pregnancy has been linked to the development of non-immune hydrophytalis, fetal death, spontaneous abortion, and severe anemia in utero. B19 is a non-enveloped virus. The icosahedral capsid is axomately and is composed of 16 structural subsonites. The B19 genome is composed of a single strand of DNA. B19 shows a markedly strong tropism for erythroid precursor cells, which might largely explain its clinical manifestations. After inhalation, B19 travels to the bone marrow via the lymphatic system, where it targets erythroid progenitor cells. The glycophospholipid glucoside, also known as the B antigen, has been identified as a key membrane component necessary for B19 internalization. This figure shows the bone marrow aspirate showing pyrovirus infection erythroid precursors, and the image of A, B, C, D showing marked erythroblast lineage of hypoplasia. Additionally, there is atypical hypobasophilic giant cells with very large prominent nucleoli. These are giant cells, also called giant chromoblast, are pathognomonic of parvovirus B19 infection. B19 life cycle. B19 affects human erythroid progenitor cells. The virus first interacts with globoside. The virus is endocytosed and somehow escapes the lysosomal route and enters the nucleus. Through strand displacement, DNA is packaged into capsids, which are transported back to the nucleus. After multiplication, the variants are released through cell lysis. B19 affected and implicated in erythroid progenitor cells in the bone marrow. The depletion of affected EPC represents the pathogenic mechanism of some hematological B19 associated diseases. Following a primary infection, the virus can establish lifelong persistence in several tissues. The infection causes anemia, hepatitis, and myocarditis, which increase the risk of either failure and non-immune hydrophatalis. Main immunological, hematological, and diagnostic events during B19 acute and persistent infection. The acute B19 infection is connected with high viral load due to the extensive replication, coinciding with a profound drop of hemoglobin value and disappearance of the reticulocyte from the peripheral blood. The virus is easily detectable by real-time PCR or conventional PCR. Up to 5 days after the acute B19 infection, anti-IgM appear and are detectable by enzyme immunoassays. After acute viremia, lifelong immunity to B19 is established consisting of IgG antibodies. They are detectable during an individual's lifetime and have protective functions. The major diseases caused by pyrovirus B19 are fifth disease, arthropathy, transient aplastic crisis, persistent anemia, and hydrophatalis and congenital anemia. Pyrovirus B19 transmission and clinical manifestation. The transmission of B19 occurs mainly respiratory drops, but it can also be spread by contaminated blood, organ transplantation, and vertical transmission from mother to fetus. Pyrovirus is widespread infection that may affect 1 to 5% of pregnant women, mainly with normal pregnancy outcome. The prevalence of infection is high during epidemic between 3 and 20, with maternal seroconversion and 3 to 24%. The B19 infection follows a seasonal variation with a high prevalence in temperate climates around late winter to early spring. Parvovirus B19 is a common human pathogen, and more than 50% of the population will be infected during adulthood. Although it occurs worldwide, the prevalence of B19 infection varies by age and geography, with high prevalence in developing countries and lower prevalence in isolated communities. In terms of age, approximately 15% of preschool children, 15% of adults, and 85% of elderly are seropositive for B19. Pregnant women who don't have antibodies against the virus are as susceptible to parvovirus infection as any other immunocompetent adults. Between 34 and 65 of pregnant women are not immune to B19 at the beginning of pregnancy, and the incidence of seroconversion during pregnancy is estimated to be 1 to 15 in endemic period and up to 11 in the epidemic period. Parvovirus B19 and pregnant women, fatal effects of parvovirus B19 infection. 13% of primary B19 infection in pregnant women results in fetal infection. The risk of maternal transmission varies according to gestational age. Pregnancy does not appear to affect the course of the parvovirus B19 infection, but the infection can affect the pregnancy and especially the fetus, as described below. Spontaneous abortion, the rise of spontaneous abortion decreases with the gestational age. At diagnosis, with a rise of 13 before 20 weeks gestation and after 20 weeks gestation. The reason for this difference remains unclear, but the largest study suggests that it may be related to multiple organ damage, which can occur in the absence of anemia or eye drops, the most classic finding associated with fetal infection. The most obvious manifestation of congenital parvovirus infection is fetal eye drops. The risk of eye drops is directly related to the gestational age. When maternal infection occurs, if the infection occurs in the first trimester. The risk of eye drops varies from less than 5% to about 10% in the infection. 13 and 20 weeks, the risk of eye drops to 5% or less. If the infection occurs after 20 weeks gestation, the risk of fetal abort is 1% or less. Congenital anomaly, currently there does not appear to be any evidence that parvovirus B19 infection increases the risk of congenital anomaly in humans. Throughout there have been case reports of coincident central nervous system, craniofacial, musculated, and eye anomaly. Studies of the long-term effects on children of maternal B19 infection suggest most infants do not have long-term adverse sequelae, but further research is needed. B19 infection itself, in the absence of eye drops or significant fetal anemia, does not seem to cause long-term neurological morbidity, but severe anemia and fetal eye drops may be an independent risk factor for long-term neurological sequelae. This figure shows the eye drops fetalis. About maternal diagnosis, systematic screening for B19 infection is not recommended, and diagnostic testing is reserved for women with high suspicion of acute infection or known exposure. Laboratory diagnosis of B19 infection during pregnancy is mainly based on EGG and IgM antibodies detection tests. IgM antibodies are detected early by the end on the first week of infection and can persist for approximately further days. IgG antibodies are markers of past infection, can become positive a few days after IgM and remain positive for years. Patients with IgG positive, IgM negative serologic test results indicate previous viral exposure and possibly immunity, suggesting that they will unlikely contract the disease in the future. In the conclusion, there is no recommendation for universal screening of pregnant women during prenatal care for parvovirus infection, but fetal identification for the correct management of affected fetus has been shown to be a strategy for improving prenatal outcome. As most pregnancies infected with B19 have a favorable outcome, indication for invasive prenatal diagnosis are rare and only indicate if there are defining signs of fetal anemia or eye drops. Fetal infection can be diagnosed using PCR and amniotic fluid, but this method is limited because it may only be positive during the very period of the disease. My presentation is coming to end, and thank you for your attention. My presentation is coming to end, and thank you for your attention.
briefly introduce myself. My name is Andriele. I'm from Brazil. I currently live in Minas Gerais, and I'm a medical student. And I wrote the article, The Complications of the Breast Aesthetic and Reconstructive Surgery. Next, please. Produce. The surgery is indicated for patient who's increase the size of the breast augmentation, and have a mammosplit reduction. Is the surgery indicated for women who has a large and have breast, which the ends interfere with the health, posture, and aesthetic of your body. Generally, woman who has the profile of the surfer from back, shoulder, and neck pain. And the same case, the trunk may even curved, which favors the development of spinal problems. The mastopexy is the surgery that aims to reverse the natural dropping of the breast and repositions the areola and stains skins, raising the breast to the original position, provide the symmetry. And the post uses tissue treatment techniques and throw breast implants. Next, please. Materials and methods. These studies consist a systematic literature review article with meta-analysis performed in the described way. Articles with the good electronic databases were included, and following were subsequently extracted, author and year of the publication, number of the patients submit to research, follow-up time, upload methodology, and results. The search the databases PubMed, UpToDate, and Cronin. A total of 16 articles were analyzed, limited in the date of publication. Between 1995 and 2022. The articles were selected because analyzed the complication and related to reconstructive and aesthetic breast surgery. Next, please. Development rates of radiation-related complication are rising among women undergoing implant reconstruction. And autologous tissue resist radiation-induced tissue damage better than implanted-based reconstruction. The late reconstruction with an allogous flap generally provides better aesthetic results with fewer complications. The transferring, not irradiation tissues, the maxtomic The site may prevent some of all the healing complications associated with radiation. They use the stack scrap. It's the solution to increase amount of the skin available for autologous reconstruction, and the act of the smoke is the negative effects to blood and supply. Next, please. The breast reconstruction in the infection is after the infection is the after prosthetic and autologous reconstruction can put the break at risk uncommon, the less than fewer 5%. Complications are begin related to site the donor tissue. It's common complication of the breast reconstruction with implants, and extend include capsular contracture or implant failure. This the prostate is reconstructed. The prostate breast reconstruction, this is the use of the acellular dermal matrix, is the role, can reduce the insides of capsular contracture, the double capsular. Implant failure leads to discriminant is breast volume with the patient notice that the breast gets smaller in a few days. The flexion is expanders of tissue or saline implants. The next, please. The other article is ruptured silicone gel implant should be removed due to the possibility the gel material causes inflammation and others reaction, especially in the gel material spread began to the breast capsule. Some implants have chaos Asia syndrome, induce autoimmune inflammatory syndrome by advanced have been reported. Breast implant disease represent a constellation of the symptoms that include autoimmune agent disorders such as rheumatology arthritis or John's disease. Breast reconstruction outcome in relation to time. In the one, 86%. The patients report accepted appears 2 years later compared to 54% in the 5 years. The texted implant had been linked to an increased risk of writing and dumpling. Next, please. And other, in the same article, post-operative breast asymmetric is the more common after unilateral reconstruction when you compare to a bilateral reconstruction. And especially after 9 sparing mastomic. Next, please. Discussion. Now, mastectomy reconstruction outcomes consortium study includes a few 2,000 patients, and the types of reconstruction using implant, explant implant, and flap reconstruction. And late reconstruction, the incidence is 5.7%, and after implants, and the 13.9% after reconstruction autologs. The overall rate of the complication was the 32.9%, with operative complications occurring in 9.3%. And the infections of the hearts were observed in 9.8% of the patients. Pass, please, for me. The patients over the age of 65 may have an overall increased risk of complication compared to the young patients. And they're probably related to medical comorbidity, but they does not appear to prediction of reconstructive failure. And now other article is perpetual to reconstruction, which is in innovator, in which the tissue span or implant is placed above the pector's major muscle. Immediately placed two of the preptor tissue explant with the subpector plexant. There was the significant difference in overall complications. In a review of the 218 women after total mastomic with skin preservation and implant reconstruction, 89 had undergone previous radiotherapy, and 133 received the post-mastomic radiation. Next, please. The patient in radiotherapy is directed to reconstruction to the staged implant, can provide an adjuvant for staged reconstruction. A retrospective review of 230 patients compared outcome after immediate reconstruction with the tissue explant, for include prepto position with the radiotherapy, prepared with the radiotherapy, and submuscular position with the radiotherapy, and submuscular position without radiotherapy. The most common complications with the capsular, it's conferring infection and seroma. Seroma is the accumulation of the fluid that can arise as and results of any surgery procedure, and during the healthy process, the hinders the definitive results of the surgery, less than 2%. Please, next. Discussion. Okay, in the latter review of the National Surgery Quality Improvement databases, in the study, the body mass, the index, the 3% remains association with the risk of the significant increased complications compared to non-based patients. And the only study compared outcomes and complications will in the 133 patients with the obesity. The study that evaluations 202 flaps infections was observed in the 3.8% of the flaps, the train, and 2.4% of the TF flaps. This is included the Braxton or abdominal donor site. The presence of the radiation is the context of the latissimus dorsiflap, and implants of the not appear to increase the incidence of infection. And diagnosis of implants or tissue explain their infection to requires a treatment. Some redness and swelling are normal and expected to inactively post-operation necessitate a careful evaluation. Next. Conclusion. Reconstructive surgery of the breast after total or partial mastectomy may be performed with the autologous tissues or breast implants, which entails a set of complications. Cosmetic breast surgery includes augmentation, reduction, and mastectomy, and the performance using prosthetic devices of tissues, regment techniques. The most patients are successful with the results, their positive results. The risk of the same complications increase for certain patients factor, smoke, obesity, and others associations with the breast cancer treatment. Example, the radiotherapy. And complications, associations with the breast reconstruction with the flap or implant that may or may not lead to operation can be classified as complication with inherent to the surgery and common to all, including seroma, bleeding, and hematoma, skin necrosis, and others examples. Pass for me. Okay, I'm finished. Thank you so much for
topic is anti-cancer effects of some medicinal plants. And the medicinal plant here we are using is Lea indica. This belongs to angiosperms, eudicots, rosids, and the family is Vitaceae. It is found in Southeast Asia, Indo-Malaya region, Western Ghats of India, Indochina, Australia, New Guinea, Malaysia, Southwest Pacific, and stem, leaves, tree of the tree are of medicinal value and are used in post-funeral rituals. Flowers pollinated by insects and the powder of flower and leaves are of potent anti-cancer effects. If given 1 teaspoon daily to the cancer patient, they act as potent anti-cancer agent. Plant is derived anti-cancer agent. It is safe. It is of low cost, fast working from root. It is simple. It is eco-friendly. Allopathic medicines which are used are cyclophosphamide, cisplatin, carboplatin, doxorubicin, oxaliplatin, melaphalan, and irinotecan. These are all medicines having negative effects on the body, and they are all having harmful side effects. But the powder of leaf and flower of these plants, they do not have side effects, and they are potent anti-cancer agents. In the cancer patient, the dried powder of leaves and flower given 1 teaspoon daily to the patient, these plants reserve are noble biochemical compounds, and more than 25 million people are suffering from cancer. Other than breast cancer, more 28 types of cancers are there, like myeloma, esophagus cancer, Hodgkin's lymphoma, non-Hodgkin lymphoma, larynx cancer, bladder cancer, lungs cancer, small intestine cancer, pancreas cancer, thyroid cancer, oral cavity cancer, rectum cancer, anal cancer, endometrial cancer, ovary cancer, cervical, liver, and so on. Ayurvedic medicines or plant-originated medicines and allopathic medicines are used, plus chemotherapy to cure cancer. But allopathic medicines and chemotherapy, they all have negative side effects on the body. And the plant-derived medicines, Ayurvedic medicines, they do not have side effects. They are easy to use, they are low cost, they are safe, fast working, they work from root, very simple and eco-friendly. But allopathic are not eco-friendly, they are not fast working, do not affect from root, or they are of high cost. Also, they are not safe and they have many negative and side effects. Other potent anti-cancer plant-originated agents are turmeric tea, wormwood, frankincense, epipodophylotoxin, Nigella sativa, Simicifuga, Fetida, Astragalus, Cytosus, Ashwagandha, moonseed, heartleaf plant, Tinospora cordifolia, Texas baccata, Culin, Corylifolium, Smilex zeylanica, Myrtus legium, Glyriza glabra, Euphorbia hitta, periwinkle plant, Docus orientalis, Artemisia, cashew, Pythoxylobium dulce, gingiber officinale, garlic, saffron, and so on. These plants, they are noble and medicinal plants. They are gift of nature for human being. We should use these Ayurvedic or plant originated medicines. Thank you. Now, next references. Thank you very much.